So this is kind of like, when your parents ask you what you're going to do for a living or something like that, what do people ask you when you go onto an airplane and you tell them what you do? So I say, oh, I'm a professor at the University of Missouri. And then, oh, that's the one in the headlines a lot, right? Yeah, that one. So what do you teach? And so over the years, I've developed less snarky answers to this. My snarky answers were, oh, well, I teach one class a year. What do you think I do with the rest of my time? Or I run a multi-million-dollar small business. What do you do? Or if I paid you $100,000 a year, do you think that's a good job? No problem. Just go out and make $500,000 a year. Or, this kind of thing. So I've decided to be less snarky over the years and now try to engage the public on what scientists do for research. And I failed for 10 years, so I'm going to take you through a couple versions of my failure. So first thing I said was, I work on polyploidy. Okay. Sleep. And then I said, I work on hybrid vigor and heterosis. It's Midwest, it's maize, everybody's interested in hybrid vigor, heterosis. That only goes so far. But then I find, well, everybody eats vegetables. So surely they must know something about broccoli, cauliflower. No, that didn't go anywhere. So finally, 3 or 4 years ago, I hit on this. And oh, no. Okay, well, we'll see how that happens. Otherwise, I'll redo it. So finally, I learned to say I work on the dogs of the plant world. So I said, well, you know how a Great Dane and Chihuahua are a dog, but they're one species, right? Did you know that broccoli, cauliflower, kale, kohlrabi are one species, Brassica Triplication? Or did you know that turnip, Chinese cabbage, and bok choy are one species? And then we talk for 2 minutes on plants, and then we talk about their dogs for a half hour. So it's great. And so then you're just being the likable professor instead of the ivory tower professor. Okay. If I want to kill the conversation on the airplane, it's no problem. I just say I'm an evolutionary biologist. And that completely damps it down cold, at least in the Midwest. So let's talk about what we mean by evolution. So of course, we're familiar with the paradigm of evolution being by slow and gradual steps. And in this talk, of course, talking about macro mutation and polyploidy and whole genome duplication, we're going to not vilify, but make Goldschmidt a hero in this case, talking about these big mutations. Of course, if you're used to doing genome scans and looking at the whole genome level, we mostly model mutations in terms of nearly neutral theory. But if you're like me and you like to collect old books, you definitely own this book. So Susumu Ohno is the one who not only coined the term junk DNA, but he's the one who originally hypothesized that you and I are actually polyploid, if you go back far enough. So just by looking at isozyme gels, he theorized that all vertebrates have this 1R and 2R whole genome duplication event. And so the punchline for my talk today, I'm going to talk about this a little bit less than usually, is this. So I go to, like Stephen, I probably go to a lot of meetings where I talk to computer science and bioinformatics people, and they tend to just treat all duplicate genes the same. Or if you read Mike Lynch's book, he has gene and genome duplication in one chapter. And I'm just going to tell you, there's a huge difference between gene duplicates and genome duplication, because when you duplicate the whole system, you have lots of more evolutionary opportunities. So in my talk today, I'm going to talk about this strange mutation, polyploidy, or whole genome duplication. I'm going to do it across a couple of time scales. So I think, along with Doug and Pam Soltis, I'm one of the few people that actually studies new polyploids, or neopolyploids, including ones we make in the lab, resynthesized polyploids, or experimental evolution, and then mesopolyploids, which is kind of what Brassica is, things that had polyploidy 10 or 20 million years in their history, and then ancient polyploids. And if you're interested in things like synthetic biology, or copy number variation, or pangenomes in humans, or primates, or other things, these duplicates are actually quite important for a whole bunch of things, including diseases like Alzheimer's and so forth. So I'm going to start bridging some work from 2004 to last year on Brassica napus, which is a canola, recent polyploid between rapa and oleracea. And then I'm going to talk about some unpublished work where we've looked in Brassica rapa about the subfunctionalization of genes that can happen, because Brassica is basically a triplicated version of Arabidopsis. And then I'm going to finally talk about a paper we published last year on macroevolution and ancient polyploidy, and then talk about the future. Okay, so just to make sure we're on the same page, when I say polyploidy, people historically have defined them by chromosome pairing, but today we usually talk about auto and allopolyploiding based on their origins. So an autopolyploid is when two genomes merge that have either identical or very similar ancestries, as contrasted to allopolyploidy, where you have two different species, in this case A and B, which join their genomes and merge. And it turns out the more we look into ancient polyploidy, that we can tell most of them are, in fact, allopolyploid events. So that's what I'm going to discuss today. So I'm not going to engage the whole debate about whether polyploidy has given the rise to diversification of large lineages in this bigger debate, although I will give an example. I think that works. So on the one hand, all mutations, including polyploidy, is bad. Right? You're mostly dead. But when it works, you have opportunities for novelty. So let's just think about that. So right, in mammals, we have sex chromosomes, and so there's not a lot of polyploidy, or almost none, in mammals. And the one good example in rodents was discredited many years ago. But that doesn't mean all animals are not polyploid. In fact, most lineages of animals are polyploid, including amphibians, insects, and we're just finding more and more, fish, many others, many flowering plants, as I'll talk about during this talk, and a lot of the other green plant lineages. And I guarantee you that you had a polyploid today. So, right, all flowering plants, including this coffee here, are ancient polyploids. Potatoes are autopolyploids. Wheat's a classic allohexaploid. Cotton. And then if you're an ecologist, you really care about polyploidy because things like Spartina are nothing more than polyploids from the Atlantic and from across the Atlantic merging together to make really noxious weeds in tidal marshes. And a lot of polyploids are invasive weeds. So this was the first experiment I did as a postdoc. So my PhD work was in the molecular systematics of monocots. And then I moved into Brassica as a postdoc. So this is when I started making crosses. So this is when I took Brassica rapa, which has the A genome, and crossed it to Brassica oleracea, which has the C genome, to make an F1, which is sterile. And then you can double the genome. Sometimes it happens simultaneously. Otherwise, you can give it a toxic shock through colchicine or whatever, which disrupts the spindle fibers. And you can get a fertile allopolyploid, Brassica napus. Now, this experiment, in contrast to the next one I showed you, I made one polyploid, Brassica napus, one plant. And then from that plant, I made pools of populations that I selected for several phenotypes. In this case, I'm showing you early flowering time and late flowering time. So after 6 generations of single seed descent and selection, I basically had 2 species, right? This plant had already flowered and was done and setting seeds just as this one was starting. So what genetic or epigenetic mechanism do you think caused this difference? Any guesses? Gene loss? Okay, good. Anybody else? That's not bad. All right. So, yeah, kind of, why, how? So you can have, this is heritable, and it's fast. And then I also want to think about how do you get these new traits, and what are their effects over time? So this is a mechanism that you probably haven't thought about if you don't think about polyploids. So the mechanism is homoeologous recombination. So I'm going to draw a picture. So here in green are a pair of homologous chromosomes, Brassica rapa, and then yellow are homologous chromosomes of Brassica oleracea. And when you put them in together into the allopolyploid Brassica napus, you now have homoeologous chromosomes. So see, you got to add some vowels here, E and O. And when you add vowels, that means chromosome 1 of Brassica rapa and chromosome 1 of Brassica napus can also pair with each other, as I show here. And this happens all the time. And you get multivalence, and then you don't have, these are not non-reciprocal exchanges, but you can have non-reciprocal inheritance, right? So reciprocal exchanges leading to non. So you can then have, let me put a phenotype on here to make this case. So what I showed you before was Brassica rapa had early flowering time, indicated by those big circles, and Brassica oleracea had late flowering time, indicated by those little circles, say 15 days of flowering and 45 days of flowering. And then Brassica napus together has a midpoint value, say 30 days of flowering. But you have this one chromosome rearrangement, and you can have all early or all late. So you get instantaneous ecological change. And by the way, there are 3 loci, like 3 places that have these flowering times. And of course, right next to each other, you have genes like FLC, Constan, Fry. They're all in a row, that, if you know your Arabidopsis flowering time genes. So when we published this, this is when everybody was going off about speciation genes and blah, blah, blah. And so we jokingly called this speciation rearrangements, right? You can get these phenotypes by, in a polyploid context, by just chromosome exchanges. Okay, so this was about the time that Loren Riesberg was becoming famous for the experiment where he took Helianthus and he remade 3 hybrids. And not only were the 3 hybrids similar to each other in terms of their chromosome rearrangements, they were very similar to what happened in nature. So if you remember all the coin and ore editorials at the time for the Science and Nature papers are like, Stephen Jay Gould was wrong. If you replay the evolutionary tape, you can get the same thing. It's not all contingency. Well, it's just chromosomes that are syntenic with each other compare, and they're in the same place. It's not really rocket science. So we were thinking, well, God, if Loren could be so famous with 3, let's do 100. So I spent a year making 100 plants from scratch. I got lucky because some of them were colchicine doubled, and then some of them were spontaneously doubled, so I can answer the critics who said this is, but I only studied 50 because this was the days of RFLP blots, and I can only put 50 on. Of course, now you can do 1 gigabase sequencing for $18, so now we would just resequence them all. So when we first made these plants, you can see them up here. You have huge heterosis. So oleracea, this fast plant, is about this tall, and the rapa plant's about this tall, and then the napus plants are like twice this size. So it's just like hybrid corn. They're a lot bigger. And when I first made them, they were all really happy, 50 S1 plants. Put a C back over them, shake them, blah, blah, blah. By the time I selfed them to the 5th generation, though, they were phenomenally different. There was late flowering lines. There was early flowering lines. There was mutant lines. Like you would have thought I threw some mutagenesis thing on them. So I've now selfed these to the 10 generation lines, and hopefully I'll just sequence all these. But what we found here was, just like I showed you in that last slide, we have all these novel phenotypes, not by SNPs or indels, but by mostly chromosome rearrangements. There's also some epigenetic effects. There's a lot of gene silencing that happens, and various other marks that we're now studying in detail. But when I published this study, let's see, when was this? This was 2007. And then in 2011, we showed that indeed it was chromosome rearrangements by molecular cytogenetics. A lot of people were still critical, saying, but this is only what happens in your crazy resynthesized lines. It doesn't happen in nature. So I was happy to prove them wrong later, which I'll show you next. So this is, again, just to get the larger point. When you make a polyploid, you can have an immediate phenotype, right? Bigger plants or smaller plants, later flowering, different flowering times, up the mountain, down the mountain. This was just one line that had a rearrangement, so it was this tall and then became a dwarf plant on the late flowering line, right? So pretty dramatic things that could be selected on. And then last year, we had sequenced several sessions of Brassica napus canola. We could finally answer the question whether the results we found in these resynthesized lines were also found in naturally occurring lines. And so I'm just showing you a cartoon of one chromosome. And so here's chromosome of rapa, here's a chromosome of oleracea, and we found a ton of these little tiny exchanges where there's like a copy and paste mechanism back and forth. And we had a huge argument in this paper. Has anybody ever been on a paper where you disagreed with your co-authors about what you wrote? So this is one of these papers, right? And so they all wanted to call it gene conversion. And I said, no, I'm pretty sure that these homoeologous exchanges. So we cut the deal and said, well, the small ones we'll call gene conversions, the larger ones we call homoeologous exchanges, but whatever. So in this paper, these larger ones we called homoeologous exchanges. And we found 17 of them, 17 of these large homoeologous exchanges. And the most frequent ones were on the largest pairs of chromosomes that are syntenic to one another. So that was 2014. And so this is a cartoon of a couple different chromosomes. So what you're looking at is this chromosome 1, 2, 3, all the way to chromosome 9 and 10. And then each of these colored boxes represents one of these homoeologous exchanges. So they're literally, this is not a rare thing. These are scattered all over the place. And if you think that they're not important, well, do you guys know what canola stands for? Canadian oil, canola, Canadian oil, right? So these are things that are low in glucosinolates and uracil acids. And so these are the QTLs that have been swept. So even what we call canola is due to these chromosome rearrangements. And all these other phenotypes are also of interest for disease resistance, oil biosynthesis, et cetera. So if you work on flowering plants and you work on polyploids, which most of them are, or they used to be, I guarantee you that these are the kind of things that have gone on. So that's recent polyploidy. So now I want to go to a little bit more esoteric topic, which is ancient polyploidy. So how do we know that ancient polyploidy occurred? So when I was a graduate student, we used to say things like 70% of flowering plants are polyploid. What was the 70% based on? Anybody? It was based on, you could look at stomate sizes and fossils and guess that guard cells would be bigger or smaller. And so anyway, so there was like some really rough estimates about ploidy. And so the biggest surprise of the Arabidopsis genome was that even though it only has 5 chromosomes, there was all these regions of synteny, right? And from these, we inferred that there were at least 3 polyploid events in Arabidopsis, and nobody would have guessed that. And then we called them alpha, beta, and gamma for most recent to most ancient. Now, at the time the alpha event was thought to be, that's somewhere in the Brassicaceae, Brassicales. The beta event was speculated to be about the origin of the eudicots, and the gamma event was thought to be around the origin of the flowering plants. And all of these were wrong because they forgot that Arabidopsis is a rapid cycling plant. So, but anyway, this idea that even what we think of as diploids today, be it corn or Arabidopsis or you and I, are actually polyploids has kind of led to some thinking about, well, what does it mean to be a polyploid? What is a diploid? Or they're just, so really we're all polyploids of various stages of diploidization. So this is a bit of a dated figure now, but the idea was polyploidy is not, you no longer ask what % of flowering plants are polyploid. You really should just ask when was the last time you were polyploid, right? Because they're all polyploid. So down here, so you have the gymnosperms, and then Amborella, sister of the rest of the flowering plants, the magnoliids, the monocots, the eudicot lineages, and then I'm going to talk about this Rosa 2 lineage. So all flowering plants share a polyploid event, which you can't see in the genome, but you can see through other analyses. And then all eudicots share this gamma event. So that was discovered when the grape genome was sequenced, that there was a hexaploid event where that gamma event is. So that's what Arabidopsis has that we can see as this gamma event. After the papaya genome was published, we knew that the beta event that one sees in Arabidopsis was not in Kikarica, but somewhere up here. And then the alpha event was somewhere up there. So when I started my lab and I learned that the papaya did not have the beta event, one of my grad students, Patrick Edger, says, we're going to go find where these 2 genome events are and see what became of them. So that's what walked us into transcriptomics. So that's what we're looking for, these alpha and beta polyploid events. Okay. So again, if you think about the big picture, a macroevolutionary picture, what does polyploidy have to do with diversification and novel traits? So a lot of this is correlation, but as I've talked with you, several of you now, we can say like the 1R, 2R events that all vertebrates have, you can look up what those genes do, and they are what make our spine, right? Or make our brain. And similarly, if you look at the 3R event that the salmonid fishes have, you can see what the functions of those genes are. And the same thing with the flowering plants, right? You can see that this gamma event in the eudicots has been pretty well studied for the effect it has had on the diversification of flowers, for example. So then there's always the question of, well, how does this evolution occur? So it's kind of back to my opening slide about Goldschmidt versus Meyer. Is it things that happen really quickly? Does things diploidize really quickly, fast? Or is it kind of an even rate? Can we associate these polyploid events with anything? What can we correlate them with, be it diversity or complexity? And when I say complexity, it could be morphology, but we're also quite interested in metabolism, because secondary metabolism often is associated with gene duplication. So a lot of people have made a little, we were talking about this in Stephen Smith's lab this morning, made a little career out of charts like this, right, where they say, oh, well, all these, here's this polyploid event here, there's not very many sea squirts, there's all these vertebrates, and here's a polyploid event here, and now you have 30,000 species of fish, and the same thing in plants. So voila, maybe it does or does not correlate with diversification. So I am less interested in that question. I'm more interested in this question, which is what happens when you double a genome? So if you have a network, so we work with metabolic networks and regulatory networks. The protein-protein interaction networks in plants aren't very good. But if you have a network and then you have a whole genome duplication, you now have the possibility of many, many more interactions. And then there's, if things are under neutral selection, there's lots of gene loss. But then you can have these novel interactions that occur. So whereas a lot of people use what they like to call systems biology to really just track down a single trait, I'm actually interested in what happens when you double the system. And I don't particularly care about the phenotype. But there is a phenotype that we can chase down, particularly with metabolism. Okay. So what I'm showing you now is a Koji figure, comparative genomics figure of the grape genome on the bottom and the Arabidopsis genome on the x-axis. So if Ambarella, or the ancestral flowering plant, has a ploidy level of, let's say, 1, and then grape has a hexaploidy event, then it's got 3 times what the ancestral flowering plant would have. And then, so grape has this gamma event. And then if you want to see these 2 other events, you can't see them by counting the 5 chromosomes, but you can see them in a genome browser, right? So for each region of grape, there's definitely these 4 regions that you can find in Arabidopsis. And you can find these through just about any method, although Gordon Burley just published a method which has problems detecting one of these. But otherwise, these are pretty robust. So if you thought that was complicated, then the brassicas are polyploidy on steroids. So I've just been describing the ancient polyploidy event that Arabidopsis shares way back here. And then let's say that this was a 8 chromosome ancestor. The brassicas then had a hexaploidy event, so it went from 8 to, say, 24 chromosomes, somewhere 10, 15, 25 million years ago. And then they diploidized from 24 chromosomes down to 9 chromosomes in the case of Brassica oleracea, which includes cabbage, broccoli, cauliflower, and relatives, and diploidized down to 10 chromosomes in the case of Brassica rapa, so Chinese cabbage, turnips, and so forth. But if you look for any particular gene, you can find things that are both single copy, double copy, and triple copy compared to Arabidopsis. And then if you're familiar with the triangle of U, right, you then make recent polyploids of that. So Brassica napus is then allopolyploid and oleracea and rapa. So if you add all these up, if you're keeping track, compared to the ancestral flowering plant, the diploid brassicas that we eat from the grocery store could have 32 genomes of the ancestral flowering plant, and the canola could have 72x. Okay, so why does that matter? So now I'm going to give you a why does it matter slide. So imagine that as we're studying under our current grant, how can these duplicate genes help you survive under abiotic or biotic stress? So this is an unpublished experiment where we took Arabidopsis and Brassica rapa, and we have genomes of both of these, and we looked at them under different stresses. So Pat Edger stayed in the greenhouse all night long and sampled plants every 4 hours, and we had 3 light treatments, 12 hours light, 12 hours dark, 24 hours light, and 24 hours dark. And then in the light dark treatment, we also had stresses. So we had hypoxia or flooding, salt stress, cold stress, and then methyl jasmonate, which simulates insect herbivory. So I'm not going to throw a bunch of hairball diagrams, but basically in other papers we've already published the Arabidopsis interactome, combination of the metabolic network and the regulatory network. And we've also not published it, but done the same thing with Brassica rapa, oleracea, and napus. And so when you think about this, the Arabidopsis network is potentially completely triplicated, but as the Brassica genome has diploidized, one genome has maintained something that looks like Arabidopsis, which we call least fractionated or subgenome 1, and these other 2 subgenomes have lost more genes. So it's not reciprocal gene loss as you find in, say, ancient yeast. So I think somebody was asking this morning. So if you look at the Brassica rapa genome, 62% of the genes are now single copy, and 32% are duplicate, and only 7% are left in triplicate. So I'm going to tell you some stories about these triplicated ones. And then if we looked at the gene expression in the root, and the stem under flooding, and under the shoots of salt stress and cold stress, we can make abiotic network for both Arabidopsis and Brassica. So the Arabidopsis interactome has 10,811 nodes, but the ones that are expressed in these abiotic stress conditions come down to be 961 nodes. And so what's interesting is a quarter of the genes in that network turn on under any stress. So it's like they're, you stress out the plant, these quarter of the genes in that network are what turn on. In contrast, if you have a triplicated genome, what are your opportunities? So here there's not 3 times 961, but 5, 6, 7, 8, 9, more than that many genes or nodes. But what's interesting is you can now, if you will, subfunctionalize or partition what you do in this network. So there's only 2% of these nodes are turned on. So let me give you a better particular example of that. So as part of our comparative genomics work, we're sequencing dozens of genomes of the Brassicaceae, and one of our first papers was we identified 90,000 conserved non-regulatory sequences, non-coding sequences, or CNSs, and in Brassica rapa alone, you can find 65,000 of these. So I'm going to show you a couple cartoons where I'm going to have exons in green, and in yellow these CNSs. And so you can, CNSs are, of course, these non-coding regions can regulate in transcription what is going on with these genes. So here's a cartoon of an Arabidopsis gene. It's got 2 exons. It's got these yellow CNSs. And then you have a triplicated Brassica genome. In this case, there's all 3 sets of exons that are there. But you can see some of these CNSs in black have been knocked out or are no longer functional. And this is classic kind of subfunctionalization. And so you can use these. So if you wanted to study humans, you would make a polyploid human, right? And then see how genes work under duplicate genes work under different functions. So if you want to know what's going on in Arabidopsis, you can actually learn quite a bit by looking at Brassica, because you can see how these things partition and function. So here's an example. So Mike Freeling actually calls this fractionation mutagenesis. Why go out and make all these mutants when you can just study polyploidy and duplication over time? So here's an Arabidopsis gene. It's got these 6 CNSs. And then in this Brassica copy, it basically is just like the Arabidopsis one, except this CNS4 is not working. And in this one, it's got both exons working, but 3, 5, and 6 aren't working. And this gene has been totally lost through duplication. And then this CNS6 is responsive to light. So I have to put these plants under light and dark treatment. So what happens to this one under light? Well, you expect this one to work and this one not to. And so that's what you see. So in this time course experiment, this Arabidopsis gene, and we have this for the whole genome. This is just a cartoon of one gene. This Arabidopsis gene over time course 6 goes up until time course 4, and then goes back down. And then this copy of this Brassica rapa gene looks exactly the same way in the light. But this one, because it's got this CNS knocked out, is dead. So it's not working in the light. So what's it doing? It's got this extra copy. So again, this isn't really subfunctionalization because it's not partitioning under different functions, but it's allowing richer gene regulation. So when you look at the other tissues, like root and hypoxia, this Arabidopsis gene is on all the time. So remember I told you that 25% of that abiotic stress network turns on. And this gene is acting just like, in the Brassica, is acting just like this Arabidopsis gene. It's on in all the conditions. But this gene that you think is dead is actually adding more product in dark and in the roots. So you can imagine with the duplicated genes, you can then do different specialties by coding in different regions. So all the terms that we use to describe genes, be it neo-functionalization or subfunctionalization or so forth, one can now use to talk about in these networks, right? So this is not a cartoon of a subfunctionalization of a gene, but what I think of as kind of a rewired network. Okay. So the macroevolutionary story. So in this project, like I was saying when we started, we didn't know where this alpha and beta event were in polyploidy in Arabidopsis occurred on the tree. So we were basically just hunting for where these events were and placing them on the phylogeny. And so Pat Edger was the grad student in my lab at the time. He's now a professor at Michigan State up the road. You should definitely invite him to give a talk here sometime. So he did this by 2012, and we could have published the plant story back then. But about that time, Chris Sweet, who's an American, but he works in Sweden, he works on the butterflies, and he was driving through Missouri on the way to Kansas City to go to the Ecological Genomics meeting, and we were showing him this plant stuff and had the little chronogram of when the polyploidies occurred and when the glucosinolate pathways occurred. And he had unpublished a study on when the butterflies evolved the counter defenses. And they lined up and we said, okay, we gotta make this one paper. So that's what we decided to do. So that's why there's this 3-year delay here to these things coming out. So I never tried to do much with pushing papers out to the larger scientific community, but we spent a lot of time like making video and doing interviews and getting quotes from Peter Raven and all this kind of stuff. And so it ended up being on NPR, Washington Post, I got interviewed by Radio New Zealand, blah, blah, blah. But I knew I made it big when it was the opening joke of a Big Bang Theory episode. So that's when I could just drop the mic and say, okay, well, if Sheldon Cooper is trying to explain the origin of mustard to Leonard, then okay, now I know. So in this episode, he was trying to explain, well, you know, the sharp taste of mustard, where does that come from? Oh, that's the origin of butterflies. Glucosino actually got it partly wrong, but I didn't care. I was like, okay, fine, cool. Big Bang Theory. Okay, so Ehrlich and Raven, classic, 1964, coevolution. What was their argument? They were saying that there are certain relationships that co-evolve over time. So the plants are going to weaponize, and then the insects are going to develop a countermeasure, and the plants are going to weaponize, and so forth and so on. And the thing that was going to mediate that were chemicals. And so they had several examples in this paper, if you've read it, but the one that they really focused on are the brassicales, the mustards and capers and relatives, and the cabbage butterflies, or the Pires. And the chemical involved are glucosinolates, these compounds. So when you put wasabi on your sushi, or you put horseradish on your steak, or you put mustard on your hot dog, these are mustard oils. And they come in different flavors. Okay. So we drew some cartoons of this. So this is a cartoon of the happy plant. It has evolved a defense. It keeps it, all the beetles and everything, aphids, everything from eating it, including this caterpillar, which isn't very happy. So the plants radiate. But then the caterpillar evolves some clever countermeasure. So it turns out the way glucosinolates work, right, it's kind of like a sci-fi movie where like the two different color liquids and they mix them together and there's a clock ticking or something like this. So right, when you bite into the leaf, the caterpillars bite into the leaves, the different compounds mix together to make something really toxic. So the insects, not the beetles, but the caterpillars have figured out a way to detoxify this, and so forth and so on. Okay, so what we wanted to ask, since we had lined up these phylogenies of the butterflies and the plants, was, were there key radiations, key innovations? Was there diversification? Could we correlate that? Are there new traits? When did they evolve? And then finally, and this is what was the key to me, was, are the mechanisms that facilitate these key innovation? Spoiler alert, polyploidy, polyploidy. Okay. Who can I pick on here? Who got their PhD before 2000? What were you doing during your PhD? Ultrastructure. You're at the microscope a lot? Yes. George, what about you?
I was doing computation or molecular.
Oh, yeah, so you were at the computer. Who else? Yes? So Priscilla here was on the scope, yeah? Lacey, what about you? What's that? Like this? Yeah. A lot of this, right? Okay, so I just mentioned this because like we don't do PCR in the lab anymore, right? So my PhD was 3 chloroplast genes. When I got there was like radiation safety. You spent all your time in the lab, and then the last year your pieces, you put it into the computer and press some buttons in this thing called PALP or blah, blah, blah. So I just laugh when I put up what we did for this, so, right? So now you're like, oh, we throw away more data every day than we used back then, right? The other paradigm shift is we used to work so hard to get orthologous regions by PCR or cloning and whatever, and now we just throw away, we use filters until we get down to only 1,000 genes or 2,000 genes or whatever. So we did a bunch of different things. In this case, we did ortho-MCL, but we also have used Synteny and later things that we're working on now. We did a bunch of divergence times. We threw the kitchen sink out of detecting polyploidy. There's a bunch of different phylogenetic ways to do it, KS analyses. And then we had already made these networks for primary metabolism for plant cell and for secondary metabolism for a new phytologist paper. And then Chris, we did just about the same for the insect side, except he also had these functional analyses for testing the substrates in the butterflies. So this is one of these papers where, it's the standard PNAS paper, and then it's got the 200-page supplement at the end of it where all the cool stuff is. So, okay. So we actually were pretty naive when we started this project about where we thought the polyploid events would occur, because we thought, well, surely the polyploid events will be where the diverse nodes are. So this is the Brassicaceae, actually including this tribe Ethionema, which has 3,000 some odd species. And so like a lot of radiations, you have the polyploid events, and then you have these two lineages, and this the species poor sister lineage has the event, but the other one, and so does the other one. But anyway, we were guessing that this polyploid event was going to be here. And so we actually sequenced this genome, and we're a little disappointed to find that it also had this event. So we found this Arabidopsis alpha event to be here. So we thought that was going to be good publishing. And then the two sister groups you probably know, the cleomes and the capers, right? You grow cleome in your yard, capers, eat them. So these have several hundred species, so we were guessing, oh, maybe this beta event will be here. And it wasn't. So we knew it wasn't in papaya down here, but this beta event ended up being way down here. So the only thing we did was we went and gathered a bunch, unless you teach plant systematics or plant taxonomy, you've never heard of any of these families in here. So there was only a few species. So we did transcriptomes of all of these to place these two events. Okay. And then we have these on a chronogram. So the radiation of the Brassicaceae is here. And then there's another radiation here, which happens to correlate to the KT boundary. Okay. So like people had found in other studies that were being published around this time, it's not polyploidy, boom goes the dynamite diversification. It's polyploidy mutate, maybe, and then diversification is a correlation. Okay, so there's many correlation stories I could tell, but I'm going to tell you this one. So after this polyploid event, you go from what I'll call a not fancy glucosinolate that you can find in papaya to the structural class I. So these are this is the indolic glucosinolates. And then you get these more elaborate, you keep adding side chains to glucosinolates to make fancier mustard oil bombs. And then finally, with this polyploid event, you get an explosion of different kinds of mustards. So when you eat capers, you get a certain flavor, but when you eat brassicas, you get more flavors. Okay. So the diversification doesn't correlate with the polyploidy so much as with these novel traits. Okay. So one way of looking at it is this. So we made a bunch of gene family trees and also did syntenic analysis to show, here's what the I'll call it ancestral, but in papaya, this is what the glucosinolate pathway looks like. So that it starts out with some cytochrome P450s and goes all the way to the molecules it makes. And so you can imagine that there was a polyploid event. This whole pathway would have been duplicated during this beta polyploid event. And there's been gene loss, but you keep these parts upstream. And that's where you get not just phenylalanine to tryptophan. So that's the origin of the indolic glucosinolates. And we can show this in synteny and in gene families. And then this pathway undergoes another polyploid event. And again, you have gene loss, but you keep these genes around here. All right. So we haven't published this yet, but what I've done is basically gone through the entire genome of Arabidopsis and Brassica and asked which ones are enriched for duplicates, either by polyploidy or by tandem duplication. So in red here is just the glycolysis pathway. So if you have one polyploid event, you'd expect 2 copies of a gene. And if you had another polyploid event, you'd expect 4 copies of a gene. But glycolysis, TCA cycle, all these kind of housekeeping things almost always go back to pretty much single copy. Whereas the glucosinolate pathway keeps 3 out of 4 of its genes. So now instead of hunting for phenotypes, I can hunt the genome for these kind of enriched pathways. You tell me all the pathways that have more than 2 to 4 copies of their genes, and I want to go look at those. And you have a gold mine of things to then go back and see what the phenotypes are, especially if they're metabolic, or work with fungi, or with bacteria, right? So we've done this study on glucosinolates, but now we're looking at phytoalexins, or you name your favorite pathway. Okay. So what about the butterfly story? So we could have published all this back in 2012, but we waited for this. So this is what Chris Sweet had on his little road trip across Kansas City that he showed me. So he had a chronogram also. And here, so the ancestral state is these caterpillars are feeding on legumes. And then at this point in the phylogeny, they move on to the capers and the cleomes. And then here, these 2 orange places, they really diversify, and there's 2 or 3 separate radiations onto the mustard family. And it was great because nature's provided a good control group. We don't even have to go to the out group. We have these lineages in black as controls because they move back onto mistletoe. So they could have evolved to glucosinolates, but now they no longer have to. So let's put it all together. So this is what we did by paper. We basically lined up our phylogeny and their phylogeny on this chronogram. And, okay. So here's this polyploid event. Here's the origin of the indole glucosinolates in the plants. This in the insect side is when they move from the legumes to the brassicales. And oh, by the way, so I have a really fancy greenhouse on the roof of our Genome Center, and I have a crappy greenhouse that has holes in it. I don't want to call it a crappy greenhouse. An heirloom greenhouse, let's call it that. You know, that's got squirrels and things running around. So it has like butterflies in it, and so if I watch the cabbage butterflies come into that greenhouse, they'll fly over the corn and everything else, and they'll go right to the brassica. How? Because the exact defense, right, the exact defense that brassicas emit is what they're attracted to. So they can always find the brassica plants. So these indolic glucosinates are actually a cue for where they lay their eggs. And that's where the insects start evolving this nitrile specifier protein that kind of allows them to detoxify the mustard oil bomb. So that's that. And then in green, you have this shift in the chemicals in glucosinolates in the plants from the caprase and cleomyces, and then the same radiation in the butterflies. And then finally, you get polyploid event and a new set of glucosinolates in the plants, and then the radiation of these guys on there. So it's pretty correlative, but it lines up twice, so that's pretty good. So, have there been key innovations on both sides? Yes. You have 2 rounds of polyploidy and 2 rounds of making more elaborate mustard oil bombs. Have they increased in complexity over time? Absolutely. You got, again, they're correlative, but you get these massive exchanges in species richness. And then this is the reason why we kept it as one paper, actually. The polyploidy and other kinds of duplications are what allow the elaboration of the pathway on the plant side. On the butterfly side, it was tandem duplication. So tandem duplication. So basically, this is coevolution by structural change. Okay. So as a side project with this, so you always have to repurpose your very expensive data, right? So Jean-Michel Ané at Wisconsin has been working on plant fungal mycorrhizal interactions. And so he was trying to figure out, well, why, you know, Arabidopsis is weird because it doesn't form these interactions, but all these other plants do, particularly legumes that nodulate and things like that. So we used the same data to look at the symbiosis specific genes in this, and found that exactly in the phylogeny where the plants have the polyploid event and then start evolving this elaborate secondary metabolism for insect defense is when they lose all these genes. So again, it's correlative and speculational, but it is something that we've now interested in, which is basically trade-offs. So if you go back to plant breeding, Karpchenko, who's this famous Russian botanist, tried to make the top of the plant a cabbage and the bottom a turnip. And he always got the opposite, which is very disappointing for a plant breeder. And similarly, a lot of these Brassicaceae, if you're in California and you like to look at serpentine soils or things like that, they occur on those soils, but then they don't have as elaborate defense to deploy for these secondary metabolites. So plants seem to have some limitations about what they can do. And so anyway, it's just kind of curious that both at a micro and a macroevolutionary level, there seem to be potential trade-offs. Okay, so last part, what are we doing? So I'm not sure if we can keep getting funding in macroevolution if it was just plants, but there's a huge space. Tim and I were just at this DOE meeting where they're very interested in how microbes and fungi evolve with plants. So if we can create these metabolic networks for the fungi and the microbes and the plants or insects bit, then I think there's a space in macroevolution to be fundable. And then the same kind of comparisons that we've done across species, we're now doing within species. So it's amazing to me that nobody's used the Arabidopsis 1001 genome data to look at any of this. So we're now looking at copy number variants relative to metabolism in Arabidopsis. And a lot of people in my lab are very interested in making a better broccoli or making a better kale. But recently, microgreens have become very hot and trendy. Like even Disney wants to sell like microgreens like that look like bunny ears or that are red or whatever. And you can get different phenotypes by growing them under different light conditions. So let's say you want your favorite arugula in your salad that's orange or red or green. You can actually make that by different conditions. So we're starting to get into a little bit of that. So I told you when I get on an airplane, I tell people I work on the dogs of the plant world, but I haven't given you anything about the dogs of the plant world. So let me give you a little flavor of dogs of the plant world. So I have these 3 graduate students working on this. So Sean is a new grad student from Florida. He's working on, along with Mackenzie Mabry here, they're working on the phylogeography and the domestication of these guys, which amazingly nobody has done yet. So we're sequenced a bunch of these. We've also looked at all of these. And Hong An is a visiting student from Wuhan in China, and he's working on all these. So after Canada, Wuhan, southern China is like ground zero for canola work. And this is something that we're doing with a grant with Mike Barker. So we're looking at the relationships among these different vegetables. So if you dig in the literature, about every possible evolutionary scenario has been hypothesized. Oh, they evolved in the Mediterranean with turnip and then went to Asia, or they evolved in Asia and then came back to the Mediterranean, or blah, blah, blah. So you have turnips, pak choi, Chinese cabbage, Brassica rapa, wild types of Brassica rapa, and this weird thing, sylvestris, which people often thought was wild. And then Trilocularis and Dichotoma are yellow and brown sarsen from India. And so we've started using transcriptomics to look at PotGen. And so this is just a quick teaser for this. So for 143 samples, so we can grow 500 and some odd reps, so 143 samples times 4. We do transcriptomes on everything, metabolome, proteome, scan the leaf, huge $50,000 experiment, and we can come up with this. So we can do de novo assemblies, and then we can map them to either the rapa or oleracea genomes, but basically we have a rapa group that looks pretty solid. A surprise, this sylvestris group is not wild. It's over here, it's a re-domestication event. And then the yellow and brown sarsen are basically indistinguishable. And then we're doing some work on how these Chinese cabbages and pak choys evolved together. So this is some early work, but the published work has like hundreds of SNPs, and this is like 28,000 SNPs, so we feel pretty confident about where this is going. And that's work with Jinhua Shi and Mike's lab. And then I'm not going to talk about it today, but I have another graduate student who's working on C4 photosynthesis. So you have bundle sheath and mesophyll cells that have separated function from C4 versus CAM, which has evolved independently multiple times. So Jacob Washburn is doing transcriptomics of both the phylogeny of the millets and relatives as well as the function of these things within there. And then finally, these 2 students, Sarah Unruh, Andrea Ravello, have I try to get them excited by an orchid project because my PhD was in asparagales. They've decided to make our lab a mycology lab. So part of the lab now has fungal plates piling up everywhere, which is how I met Tim the other day. So I challenged them to write a grant to fund their work. So they're now funded by JGI to sequence 400 members of these, of Tolosnella, Ceratobasidium, and Sebacina. And they're fantastic because they grow in plates. You can grow them with or without the orchid. You can grow the orchid with or without the fungus. You can grow them all by themselves, so you can just throw omics at it. And it's pretty cool. So just alpha, if you don't know about orchids, orchids don't have endosperm, so they don't have like their baby brother to eat when they grow up like other flowering plants do. And so they need this fungal partner, but the fungus doesn't need the orchid. And so why the fungus is in this relationship is a really basic question. All right, so just to summarize, so don't let anybody tell you that polyploidy is not rampant across life, because you and I are polyploid, if nothing else. Polyploidy allows right away an evolution for massive changes in phenotype, even if it's by those homoeologous exchanges I was telling you about before, and other ways to vary copy number variation. These new recent and ancient polyploidy, we've documented these new traits. So you can wave your hand about correlations or whatever, but absolutely these duplicate genes are doing things. What I like to call systems biology, and again, I refrain from showing you lots of hairballs, but you can find some pretty deep insight by letting natural gene loss be a natural experiment for how you can test function. And again, I didn't talk about it today, but it turns out if you look at the human embryo and cancer, there's more in common between those than human embryo and other cells. And so because of the Warburg effect and other things, you can instantly think of polyploidy for human health, particularly cancer. So we're looking into that on the side too. So I've been generously funded by NSF for the last few years. I'm still trying to break into NIH, but maybe George can tell me how to do that. I don't know. And then we've had 2 projects funded with DOE to sequence 20 some odd Brassicaceae and now 400 orchid fungi. And this is my lab. I have 1, 2, 3, 4, 6 grad students and some amazing post-undergraduates. I think this one interviewed here at Michigan, Wade. And yeah, that's it. Thank you. George, you have a question.
So you said early in the talk there was an experiment you made polyploids in the lab and you selected for early flowering.
Yes, correct.
Do you have a control that is a diploid? You do this.
No, that would have been a better experiment, huh?
I wonder whether the polyploids evolve faster in the selection, they respond faster.
Yeah, so that's a good question. And now I feel like I have a better experiment to do next time. Yeah, so I think Jonathan Wendell's done that experiment comparing molecular evolution of diploid versus polyploid genes, and the polyploid genes do evolve faster.
In terms of this adaptive change.
Yeah. I don't know, what do you, I'm going to go with your suggestion, you tell me.
I think it would be interesting to check.
Okay. George says it's interesting to check. Next. Oh, come on. Yes.
So I think if I understood a slide that you showed.
Which by? Mm-hmm.
When polyploidization takes place, it looks like with respect to some of your biochemical pathways, there's a bias in terms of what genomes are.
Yes. We call it biased fractionation.
Right. And so are there any general properties that are there any general properties that have been discovered with respect to that?
No, it's an open question. So there's some hypotheses. So Mike Freeling has hypothesized, because he's a transposon guy, the skinny genome wins. Okay, so we're talking about allopolyploidy, 2 separate species. So if you have one fat genome full of transposons, the skinny genome wins because all of its RNAi machinery can target, has all these things over here to target and knock out for the transposons, where this person has fewer things over here. But it's definitely a genome-wide effect. So we know it's something epigenetic going on. So I have friends who are working in Mimulus who looked at that, and it's not methylation. And so I'm going to work with Blake Myers on some small RNA work. There's some places that we're wanting to look for that, but we do not know the mechanism. And we also, with anything with polyploid, we never know if we find is it a special case for Brassica versus, so whenever I give a talk like this, the cotton people say, it doesn't work like that in cotton, or it doesn't work like that, but I think there probably is a general explanation for that, but we don't not know, and nobody I know knows. Although the labs in China are sequencing everything like crazy, so they might be the first ones to find out. Yeah. Yes?
Birds and mammals are boring relative to others.
Very boring.
In the context that you can't really have polyploids,
Yes.
homoeologous,
Right. Yeah.
napus, etc. When you look at and you, if I remember correctly, ascribed that to Resynthesized chemistry.
I put that out there as one of many theories.
Which is the most plausible theory?
I don't know.
But when
Do you have one?
I'm asking you that.
Oh, okay.
So when we look then across, say, plants, are there places of plants which also are less flexible in the genetic?
Yeah, okay, good question. So it's something Mike Barker and I are working on again. So there's dioecious in plants, right? So there's no huge clades of flowering plants that have, like there's no family of flowering plants that's dioecious, but there are certainly lineage like, oh, this asparagus or this, right, independently evolving. So we've tried not very hard, but we've tried to make polyploids from these. And we've also have looked at where polyploid events occur and where there may be some flexibility in the sex determining system. And again, this isn't my work, but Mike's looked at this with insects. So in insects, there's sex determining things, and there's now some new evidence that polyploidy is actually rampant, and so people are looking very closely at that, and it looks interesting. So that's at least testable, right? You can say, oh, extant lineage, these sex determining regions or whatever. I don't know about how in birds and ZW and all that works, that where the flexibility is there, but I can say this though. You don't know to look for polyploidy until you look for polyploidy, right? So even the fact that we're polyploid was a mystery until people really, and it was controversial for a long time, and now it doesn't seem to be, but I'm guessing that if you look pretty deeply at some current genomes that people say are not polyploid, they might be, but I'm letting other people take care of that. So if I can't even see it like on that little diagram like I put with my grape on Arabidopsis, I get uncomfortable with it. But any other questions I can answer as poorly as that one? That was pretty bad. Are you not entertained? I can whip out my gladiator sword now. Yes, Stephen.
So, you didn't talk very much about gene duplication.
No.
First you're talking about genome duplication. What do you feel for especially angiosperms, the relative importance to organ novelty?
Yeah, very good. So Ann Osborne and others have made a pretty good career out of looking at secondary metabolites coming from these little modules of things. And so I am confident that those are mostly segmental duplicates. Does that start to answer your question? I'd be very interested if I was working on Caryophyllales of like where the beta-lin pathway comes from and where the little carnivorous tank toolkit comes from, and I would not be surprised if duplicate genes didn't have something to do with that. Am I getting close?
Yeah.
Okay. Yeah. Any other dancing I can do with the... So can I ask you, what would you do with my fungi? Orchid fungi.
Well, I think it's interesting because you say that we don't know.
I don't know. Do you know?
Whether they're enslaved, like, you know, the question of whether they're enslaved or not,
Yeah.
and what benefit they can get
Yeah.
from the plant. What, if anything, characteristics and
Yeah. Yeah. But we can do it in the Petri dish, so it should be a cheap experiment.
But how do you know they are not getting anything from that?
Yeah, so we can tag the carbon and stuff and see what's going on, and we don't know. Maybe they're just waiting for the plant to die. We also know that the fungi can eventually eat the plant. So I'm really interested in the switch. When does the fungus say, you know, it's got one part of the fungus is attached to grass and things it cares about. It's got one little piece of it attached to an orchid. Is that dessert? Oh, it's got a cool micronutrient. I'll let it suck off my finger until I'm ready for some dessert. And or does it care? Or, you know, I don't know. So we're going to focus really hard on the signaling, so we have some clues about what to look on for the signaling, but it's a pretty good basic science question. And I thought it was like completely not fundable, but there's actually people who want to move that toolkit into grasses and things, and then maybe DOE will keep paying for it. I don't know. So anyway, it's a basic science question. So now are you entertained?
All right.
All right. Thank you.
Great.